It's my pleasure now to move on to our third speaker of the day. And our next speaker is Clayton Wollner. Now, Clayton is a PhD candidate in Justin Richner's lab at the University of Illinois College of Medicine. Now, his very strong interests in both the field of vaccinology and RNA therapeutics as well. And his presentation fits right into that theme. And the title is A Dengue Virus Serotype 1 Messenger RNA LNP Vaccine Elicits Protective Immune Responses in Mice. So it's my pleasure to hand over to Clayton.
Thank you so much for that kind introduction, Richard. And thank you so much to Precision Nanosystems for hosting this symposium. I feel a little out of place amongst such accomplished, well-known scientists in the field, but here I am, and I'm very thankful to be presenting the data and findings of my thesis project concerning a dengue virus serotype 1 mRNA LNP vaccine. So let's just jump right into it. So just some basic molecular biology behind flaviviruses. They are enveloped viruses. They are positive sense single-stranded RNA genomes of 10 to 13 kilobases. And they contain a single ORF that codes for, let me get my laser pointer here. Get that. They code for 3 structural proteins as well as 7 non-structural proteins, which are subsequently cleaved into their individual components. Some more notable names within the flavivirus family itself. A lot of people will recognize West Nile virus, Japanese encephalitis virus, Zika virus, yellow fever virus, and tick-borne encephalitis virus. And just quickly, I wanted to touch on the viral particle itself. Structurally, it is composed of the capsid, the precursor membrane protein, as well as the envelope protein. And those would produce, the structural components of this viral particle here. For the purposes of this talk, I do want to highlight the fact that when just precursor membrane protein and envelope protein are expressed, you get production of something called a virus-like particle, or VLP. And essentially, this is just a particle comprised of the PRM and the envelope proteins. It's non-infectious. It doesn't contain any non-structural proteins or genome or capsid protein. And this image compared to the last one is an extreme oversimplification, but the VLPs themselves are smaller with a different symmetry than the wild type infectious particles. But overall, the epitope landscape on the surface is one that mimics wild type infectious virus. So these particles are something really great to produce in order to produce a highly neutralizing antibody response. So some specifics about dengue virus itself. There are 4 dengue serotypes, so dengue 1, 2, 3, and 4. They are transmitted by Aedes species mosquitoes, predominantly Aedes aegypti, and to a much lesser extent, Aedes albopictus. And there are 390 million worldwide infections annually. And the disease states for infection can range from a mild self-limiting fever to more severe cases that involve inflammation, plasma leakage, and organ bleeding and impairment. The highest risk for these severe disease states occur when someone experiences a secondary challenge with a heterotypic serotype. So for instance, if your primary infection was with dengue 1, you would be more likely to experience severe disease if you were infected with dengue 2, 3, or 4. And this is due to a phenomenon called antibody-dependent enhancement, or ADE, which is something I will touch a little bit more on in some upcoming slides. So currently there's an unmet need for a protective dengue vaccine. There are lots of dengue vaccines that exist, a lot of which utilize live attenuated platforms. Probably the most notable dengue vaccine would be Dengvaxia, which incorporates all 4 serotypes of dengue. It utilizes the PRM and the envelope from dengue virus into the backbone of yellow fever virus. Unfortunately, after a large population of people in the Philippines were vaccinated with this vaccine, what they saw was increased susceptibility to severe disease and a secondary challenge through ADE. So an effective dengue vaccine is going to need to elicit protection against all 4 serotypes while also avoiding ADE. So before, jumping into ADE itself, I wanted to sort of prime people with a concept in the flavivirus field concerning the viral particle heterogeneity. And that is, in a nutshell, that viral particles exist in different states. And so this graphic here does a really good job of showing this. What you have is you have rough and smooth viral particles. And the rough viral particles are immature and non-infectious, and the smooth are mature and infectious. And this is a result essentially of the precursor membrane protein, which I show here. And during normal viral maturation, precursor membrane protein is cleaved by host furin into its mature form, which is just membrane protein with the PR having fallen off. But this process is not 100% efficient, and so you'll have intact premembrane protein existing on viral particles. And what this does is it keeps envelope proteins in sort of this upward configuration. And so the disadvantages of this are twofold. It's that you have intact premembrane protein, which in turn induces antibody production against intact premembrane protein. And additionally, when the envelope protein is sort of in this up conformation when the PRM is intact, it's hard to see, but these little red dots are the fusion loop. You can see them orange here, which is hard to see against sort of this pink background. But in this up conformation, the fusion loop is also more exposed, the fusion loop of the envelope protein. And in this more exposed conformation, you can have more of an induction of antibodies against the fusion loop itself. And the next slide, I'm going to sort of tell you why that is not advantageous in the concept of a vaccination or protective immunity. So again, just to remind you all what the problem child epitopes are here, that would be intact PRM and the fusion loop of the envelope protein. And you have a graphical representation here of antibody-dependent enhancement. But essentially what you have is you have existing flavivirus antibodies that are cross-reactive and sub-neutralizing. And a good example of these would be the intact PRM and fusion loop antibodies. And these would target partially mature and immature viral particles. Now, these opsonized virus experience increased viral entry in Fc gamma receptor positive cells. And this is bad for a couple of reasons. So when you have immature virus that's opsonized by PRM antibodies, that virus will be internalized by APCs via Fc gamma receptor mediated endocytosis. But within the endosome, there is actually host furin available, and that furin will complete the maturation process of PRM. So once intact PRM is cleaved, the antibodies against intact PRM are rendered essentially useless within the endosome. These antibodies fall off, and the virus can fuse with the endosomal membrane and release its genome. Additionally, if you have virus that's opsonized with fusion loop specific antibodies and are internalized into the endosome, late endosomes actually become acidic, and this acidic endosome actually induces irreversible conformational changes to the envelope protein. And fusion loop antibodies cannot prevent those conformational changes. So within an acidic endosome, the fusion loop antibodies fall off, and the fusion loop of the envelope protein can fuse with the endosomal membrane, resulting in subsequent release of the viral genome and infection of the cell. So these are just some examples of bad antibodies and how they contribute to enhanced infection. So an overall goal of my project long-term would be to create a tetravalent dengue virus vaccine using the mRNA LNP platform. So right now, I mean, this may change, but right now we're planning on possibly just creating constructs against all 4 dengue serotypes and incorporating them into a vaccine. That may change as time goes along and we discover new things about our vaccine, but that's the goal for right now. I just wanted to sort of walk you through the general concept of it here. Up above here, we have the full flavivirus genome, just for context. And below, we have the mRNA construct that we deliver to these mice via vaccination. This construct only contains the premembrane protein and envelope protein from the dengue virus genome. And typical of RNA therapeutics themselves, there are other elements in this construct, of which are the 5' and 3' UTR, a poly A tail, a 5' cap, as well as a signal peptide for proper processing of the proteins through the trans-Golgi network. Now, a major thing with dengue or flavivirus vaccine design in general is avoiding ADE. Now, in order to do this, initially, we wanted to incorporate mutations that would eliminate production of cross-reactive non-neutralizing epitopes. And a major one that I had discussed previously was the fusion loop. And looking at a multiple sequence analysis here, the fusion loop is actually 100% conserved across all 4 dengue serotypes, as well as multiple other flaviviruses, including Zika, West Nile, and yellow fever. So highly cross-reactive in that it's recognizing the exact same epitopes, but not neutralizing. So all in all, not an antibody that you want to protect yourself from dengue virus. So to counteract that, we utilize 3 mutations, G106R, L107D, and F108A, in order to prevent production of fusion loop specific antibodies. And these mutations have been previously characterized, not altogether, but previously characterized to reduce or prevent production of fusion loop antibodies. And some have actually been implicated in increasing envelope protein expression as well. So initially, we wanted to characterize protein expression of this mRNA construct in vitro. Oh, I'm sorry. Actually, quickly, I wanted to go over some concepts behind mRNA vaccine design itself. Number one is that RNA is actually a very fragile, sensitive molecule. I swear it degrades just by looking at it the wrong way. And as such, the way that you package and protect your RNA is of the utmost importance. And so my lab decided to utilize the Precision Nanosystems benchtop assembler to produce lipid nanoparticles with our mRNA inside. And this is really great for protecting your RNA during cold storage against degradation, as well as being a really great way to deliver your mRNA to cells in general. And you can see here an overview. Essentially, what you do is you just have an aqueous solution of your RNA in this case, and dissolve lipids in organic solid and solvent. And using laminar flow mixing, you encapsulate your mRNA into a lipid nanoparticle. This lipid nanoparticle interacts with apolipoprotein E, which allows it to be endocytosed by cells, where the LNP is degraded in the endosome and releases the nucleic acid, in this case, the RNA. Another thing to consider is that RNA itself can be immunogenic. Specifically with in vitro transcribed RNA, there are double-stranded RNA byproducts that are highly immunogenic. And so I think everyone here knows what RNA is comprised of. And a way to overcome the immunogenic effects of RNA itself is to utilize nucleotide analogs. So instead of using uridine, we used an in vitro transcription kit that utilizes pseudouridine as a way to avoid immunogenic effects against the RNA itself. Because ultimately, we want the RNA to produce protein, and we want the immune response to be against the protein, not our RNA. So it's very important that we avoid any immune responses against RNA when possible. So as I started prematurely before, we wanted to characterize this RNA in vitro. So right off the bat, what we did is we transfected 293 T cells with both the wild type construct and the delta fusion loop mutant construct with the 3 mutations to the fusion loop. And using 1A1D2 monoclonal antibody, which is specific for domain 3 of the envelope protein, we were able to confirm protein expression in both the wild type and mutant constructs. Additionally, we didn't see any deleterious effects from the mutations on protein expression. In fact, the slightly darker band here actually sort of supports the idea that these mutations can actually increase envelope expression, albeit very slightly. We did, however, want to confirm that we had successfully removed the fusion loop epitope. So using a 4G2 monoclonal antibody, which is specific to the fusion loop in the envelope protein, we unsurprisingly got a band for our wild type, and thankfully saw no band for Western blot of our delta fusion loop construct. So we were able to confirm that we had indeed mutated our fusion loop to the point where antibodies wouldn't be produced against it that would be effective. So I talked a little bit about virus-like particles, and that is ultimately what we were aiming for with our vaccine, and ultimately what we were thinking we were getting. But there were some things we needed to do to confirm that. So obviously, electron microscopy would sort of be the most obvious route, and we did eventually do that. But before doing that, we wanted to confirm, at the very least, protein complex expression in vitro transfected cells. So what we did is, from this experiment, we actually took the supernatant and we purified it via 20% sucrose cushion and ultracentrifugation to pellet protein complexes, and in our case, hopefully virus-like particles. And after running a Western blot of the pelleted supernatant, we were able to see that we were indeed getting protein excretion from both the wild type and the delta fusion loop constructs in the supernatant. At this point, we still weren't sure if they were virus-like particles. Additionally, we ran a GAP-DH blot to ensure that these bands were not from like dead cell contamination, for instance, during the ultracentrifugation process. And thankfully, we saw pretty much no contamination, especially compared to the lysate control. But we did eventually splurge on electron microscopy, and we were able to get some really nice images of that. This one being the better of most of them. And we are confident in saying that these are virus-like particles for two reasons. Number one, these particles are of a homogeneous size. So the particles are similar sizes to each other. Number two, they are within the correct size range. So as I mentioned previously, virus-like particles should be about 30 nanometers in size. And these are approximately 30 nanometers in size. So we were fairly confident and happy to see that we were getting virus-like particle excretion from our mRNA construct. And then after seeing our vaccine perform well in vitro, we wanted to move in vivo to see if this would work in a whole animal model. So just a quick overview of the vaccination schedule. We used immunocompetent C57 Black 6 mice, and we used a prime boost vaccination strategy with a prime vaccination at day 0, a boost at day 28, and serum collection 2 weeks after the boost. And additionally, for a positive control, we infected C57 Black 6 mice with dengue virus 1. And it's important to note that our viral stock of dengue virus 1 was actually an identical sequence to the vaccine construct we used. So in future slides, when I show you some differences in the immune response, that's not a result of any differences in the sequence, because they are actually identical. We also utilized a GFP negative control, and within this study, we also used a high 10 microgram dose of mRNA, as well as a low 3 microgram dose of mRNA. So what we looked at initially was serum neutralization. So using focus reduction neutralization tests, we were able to produce some neutralization curves from these treatment groups. Unsurprisingly, virus had probably the best neutralization curve. GFP had nothing more than background, and our vaccines, both high and low, performed really well with some nice neutralization curves there. Producing EC50 values from those neutralization curves pretty much just supported the patterns you see here, with virus being the highest, GFP having pretty much no neutralization activity besides background, and then our vaccine constructs performing close enough to the virus for our liking. And they induced equivalent neutralizing antibody titers between the wild type and fusion loop. So the overall goal of our project was to produce a neutralizing antibody response. Within this project, we really were aiming for the antibodies to be protective. We did, however, look at cellular immune response, as it is the prudent thing to do in these studies. So at the end of the vaccination experiments, we would collect the spleen and homogenize them. And we actually sent the splenocytes to our collaborators at St. Louis University College of Medicine. And they stimulated these splenocytes with overlapping peptide libraries of dengue virus envelope protein. And we were actually able to see CD8 positive and CD4 positive T cell activation within this experiment. It is important to note that this activation was highly specific, and that they also ran dengue virus 2 envelope protein and did not see activation of these T cells. Additionally, they ran dengue virus 1 non-structural protein as sort of an off-target control, and they didn't see any activation with that as well. So ultimately, we are creating a vaccine to protect, hopefully one day, people from dengue virus. And we wanted to see if this vaccine could protect mice initially. So we wanted to undertake a vaccine efficacy experiment. So this involves a mouse model, the AG129 mouse model, which is very well known within the flavivirus field. These mice are interferon alpha, beta, and gamma receptor deficient, so they don't have a type 1 or type 2 interferon response, and as such are permissible to dengue infection. And what we did is we sent vaccine to our collaborators at St. Louis University, and they vaccinated the mice with the same vaccination schedule that I outlined before. So prime at day 0, boost at day 28. And then they actually waited almost a month before collecting serum and subsequently challenging these mice with virus. But with the serum that we got from these mice, we were able to produce EC50 values the same way that we did with C57 Black 6 mice. And the AG129 mice had pretty impressive EC50 values. And just to contextualize that with the C57 Black 6, within the AG129 vaccinated mice, their EC50 was actually higher than the virus-infected C57 Black 6, which virus, I would say, would probably be the epitome of a good neutralizing antibody response, only because those are the same particles you would see during an actual infection. So we saw elevated humoral immunity in the AG129 mice, and we believe this is the result of a reduced interferon response. So as stated previously, foreign RNA, especially double-stranded RNA, can be immunogenic and lead to an interferon response that can subsequently reduce protein production in the cell overall, which obviously has an effect on antigen expression from the mRNA construct that we deliver. So with AG129 mice not having an interferon response, we expected the protein expression to be higher, and we were sort of pleasantly surprised to see that the immune response was also in turn elevated as well. So this was an efficacy study, and we did eventually challenge these mice. We didn't just look at their serum EC50 values. So just again, an overview of the vaccination schedule. But after challenging these mice, they were monitored for weight loss and survival. And we were really happy to see these results. So in the vaccinated AG129 mice, we saw greatly reduced weight loss compared to the GFP vaccinated mice receiving a control GFP construct and an mRNA LNP. And then when you look at survival, we actually saw 100% survival in our vaccinated mice as compared to 100% lethality in the GFP vaccinated mice. So pretty much the exact results we were hoping to see. We were very happy to see this. So, you know, in an immunocompromised mouse model, though, where you sort of have greater protein expression and a subsequent greater immune response, efficacy experiments aren't entirely indicative of what you would see when vaccinating an immunocompetent population. And this is actually something a reviewer asked us to do in our most recent publication, and we were happy to do it. But we wanted to isolate the protection provided by serum antibodies. And in this specific experiment, we wanted to isolate the protection provided by serum antibodies in immunocompetent vaccinated mice. So the C57 Black 6 mice. So what we did is we passively transferred serum from C57 Black 6 mice that were vaccinated into the AG129 mouse model before challenging them with dengue virus 1 West Pacific. And looking at weight loss and survival here, again, results we wanted to see. We saw weight loss or reduced weight loss in the vaccinated group as compared to having received no serum or naive serum. And as far as survival, I think this was 1 mouse out of 8 that succumbed to the infection, which was significantly better outcome for the other treatment groups of no serum and naive serum. So in this scenario, the antibodies are what protected the mouse from infection, which was sort of what we were aiming for with our vaccine anyway. So as I've stated previously, antibody-dependent enhancement, or ADE, is a very important thing to look at when designing a flavivirus vaccine. And so what we did is we designed and optimized an in vitro ADE experiment. And in a nutshell, what this is we incubate serial dilutions of serum from vaccinated mice with dengue virus to opsonize it. And we subsequently infect K562 cells, which are Fc gamma receptor positive. If you recall, that's the receptor that's responsible for endocytosing opsonized virus and are the ones that eventually experience increased viral uptake in an ADE scenario. And after 15 hours of incubation, we quantified the infected cells via flow cytometry using a dengue virus envelope protein or antibody. And just to sort of show you what a graph like this would look like, this isn't our results, but using a monoclonal antibody E60, which is fusion loop specific and therefore highly inducing of ADE, you would see a curve like this where at either end, either the antibodies are too high, where they actually are fully neutralizing the virus, or too low, where there aren't enough antibodies to enhance infection. But then somewhere in between, you see this curve where the antibodies are actually increasing viral uptake and infection of the cells. So looking at our data, this is sort of the signal we got at the end that we based this curve off of. But what this is this is the signal of dengue virus 2 envelope protein. So in our ADE experiment, we enhanced dengue virus 2 with dengue virus 1 antibodies. And we were able to create curves here of ADE. And the black is virus, and the blue and red are the vaccine constructs, with green being naive. So our vaccine constructs, both wild type and fusion loop, did enhance a little bit, but at the greatest enhancement, we actually saw only twofold infection increase compared to naive. Whereas with the virus at the highest, you actually see 8-fold increase. So we greatly reduced the ADE, at least in an in vitro scenario, with our constructs. Additionally, we did look at production of infectious viral particles from this infection. So instead of fixing and staining at 15 hours for flow cytometry, what we did is we just extended this experiment until there would be viral shedding. And then we subsequently looked at how much virus was in the supernatant of these infected cells. And it's a bit noisier of an experiment, but essentially you see the same patterns from this graph here, recapitulated here, with virus having the highest infectious viral titer, GFP having none really, and the vaccine construct somewhere in between with significantly reduced viral titer compared to virus, which is what we were looking for. Surprisingly, the wild type and fusion loop constructs performed similar, and they were similarly low, but we were expecting the fusion loop construct to have reduced ADE compared to the wild type, simply because the wild type still contains the fusion loop epitope. But this got us to thinking that maybe VLPs produced by an mRNA vaccine like this could possibly have a slightly different epitope landscape, so to speak, that maybe the proteins comprising the outer shell of the viral particle are actually presented in a way that inherently reduces antibody production that can lead to antibody-dependent enhancement. That will require much further investigation, but right now we're just theorizing that that could be the case. So just a comparison of the humoral immunity of the RNA vaccine and the virus-infected mice itself, just to sort of to contextualize everything. So this is the EC50 of the virus-infected mice and the high-dose 10 microgram vaccine administration. And these are fairly similar, but the virus had about 2 times higher EC50 than the vaccine construct. But relatively speaking, they're similar enough and both likely protective in a homotypic challenge. But when you compare that to the ADE, the 4 times higher ADE that you see in the virus compared to our vaccine constructs here, you can't account for that difference with a difference in viral titer. And we just wanted to highlight that, because some people might think, oh, well, maybe your vaccine constructs lead to a lower antibody titer overall, and that could likely be what's causing this difference in ADE. But based off of these 2 graphs together, that should not be the case. So overall, we believe our mRNA vaccine elicits a qualitatively better humoral immune response. Maybe the EC50 isn't as high, but the antibodies are of a higher quality. So just to sort of conclude this, we produced a dengue virus 1 PRM envelope protein mRNA LNP vaccine, and it elicited serotype-specific protective immune response. Future direction is that we are right now designing and characterizing constructs for dengue virus 2, 3, and 4. And we're planning to look at, cross-reactive neutralization as well as ADE across those new constructs as well. And as stated previously, we're planning on designing a tetravalent vaccine platform to hopefully get broad neutralization against all 4 dengue serotypes. And as I mentioned in the previous slide, we're planning on mapping the polyclonal repertoire of virus-infected mice compared to vaccine administered mice to see if there's an overall difference in the antibody epitope targets between the two, which might explain the differences in antibody-dependent enhancement induction. So with that, I would just like to acknowledge everyone in the Richner Lab, my advisor, Dr. Justin Richner, who is a great advisor. I mean, there's no one on this planet who could argue that he's not passionate and very excited about science every day. He definitely loves what he does, and it's infectious to everyone else in the lab, no pun intended. Our lab manager, Michelle Richner, fellow graduate students, Allie Bennett and Jake Klass, as well as our collaborators at St. Louis University, James Bryan, Amelia Pinto, and Mariah Hassert, who did the splenocyte activation experiment, as well as the AG129 challenge studies. So with that, I would like to take any questions, and thank you for listening to my talk.
Awesome. Thanks so much, Clayton. I really appreciated the talk. I really appreciated the pun at the end. That was really good as well.
Thanks.
Much like I mentioned previously to Richard, I think it's another instance where it's really exciting to see, RNA vaccines now in another space, something different, something exciting, something where we need something different, I think, and really seems promising. So you should definitely be very proud of the work you presented here today. It's fantastic.